Here in part 3, let's go back and explore now the life cycles, the alternation of generations, and the connections that now emerge between plants and animals. In angiosperms, we find that flowers are the reproductive structures. We have stamens, which contain the male sporangia that produce pollen and eventually the male gametophyte, pollen grain. And then we have the carpel, which is the female megasporangia that produces the female gametophyte inside an ovule. Let's take a break. And if this was live, we'd be asking you to come up with a single word to describe the purpose of flowers. In this diagram, we show you the alternation of generations as we find it in angiosperms. Beginning with the anther, the male organ, that through meiosis forms a microspore, a pollen grain. That's the male gametophyte, which then lands on the stigmatic surface of a female. At that point, the pollen grain can germinate and grow down to the egg for fertilization. In the female part of a flower, we have an enclosed ovule, an enclosed ovary, in which cells undergo meiosis to produce a megaspore or egg. Then we have in that ovary an egg cell and we have an egg cell and other ancillary cells that are associated with reproduction. Fertilization takes place in an enclosed part of the plant, whereas fertilization takes place on the surface in the gymnosperms. It is enclosed within the ovary in angiosperms. There is a double fertilization that takes place. One pollen grain fertilizes the egg, and the other fertilizes several other cells that will end up forming the nutritive tissue, which is a 3N tissue associated with embryo development. So what we have then is the production of a diploid organism, which ends up being a seed, which can then germinate, repeat the cycle of, again, female structure producing an ovary with eggs and ancillary cells, a double fertilization process, creating a zygote plus a 3N tissue associated with helping the nutritional development of the young plant. And after the seed is produced, we again find a 2N sporophyte. The majority of the life cycle of angiosperms is in the sporophytic phase. And this contrasts with the majority of, let's say, a moss or other bryophytes, where the majority of the life is spent in the 1N gametophytic stage. Within the angiosperms, this being the gymnosperm outgroup, we find a number of families, but two groups predominate. One is the monocots, and the other is the eudicots. Monocots and dicots differ morphologically. Monocots have a single cotyledon or seed leaf. Dicots have two cotyledons, so that when we have the initial germination event, as the seedling emerges, there are two leaves in dicots and a single leaf in monocots. If we look at a cross-section of the vascular tissue, what we find is that the xylem and phloem are scattered throughout the entire stem. Typically, the stem only lasts for a single year, a single season. Whereas in dicots, we find that the vascular tissue, that is the xylem and the phloem that we talked about earlier, are located on the outside, and with the cambial cells, cell division takes place every year, producing new xylem and new phloem. So stems of dicots tend to last multiple years. When we look at the vein structure of a leaf, in monocots, the leaves tend to be parallel, the veins tend to be parallel, whereas they tend to be branching and net-like in the dicots. Two contrasting vascular structures and vein arrangements. Animal coevolution with plants occurs at 3 different stages of the plant's life cycle. We find that there are plant-animal interactions associated with pollination, that is the transfer of pollen grains from one plant to another. Once a fertilization event has taken place and there's a new seed, we find plant-animal interactions evolving with seed dispersal. And lastly, we find in the adult sporophyte patterns evolving that reduce or limit herbivory, that is the consumption of plant material by animals. So we find 3 different phases in which we see the evolution of plant-animal interactions: pollination, dispersal, and herbivory. Let's consider some of these examples of coevolution. Plants are incredibly smart, so to speak, and they're able to convince animal systems to transfer pollen from one flower to the next. This is much more effective than broadcasting the pollen grains, as happens in gametophytes, or releasing them into water, where they may or may not find another gametophyte to fertilize, a female gametophyte to fertilize. So the evolution that we see in angiosperms is targeted movement of pollen grains, whether that's with the butterfly in the zinnia flower, the hummingbird in this hibiscus, or a cactus flower in bats. Similarly, once reproduction has taken place and we have the formation of a new zygote and eventually a seed, plants have evolved mechanisms to disperse those seeds. Sometimes it's in the form of a fruit, sometimes it's in the form of a reward. But plants have developed mechanisms to convince an animal, to come disperse the seed. There are many kinds of pollination systems that have evolved. Some of these are really quite hard to believe. One we'll talk about in more detail is the carrion flowers. These are flowers that smell like poop. They attract carrion flies, and in the process, the carrion flies end up transferring pollen. They attract hummingbirds and other kinds of birds with a nectar reward. So in the process of sticking its beak in to get the nectar, the hummingbird picks up stamens. Some plants attract bees with a small nectar reward using bright colors, usually colors that are visible in the ultraviolet wavelengths. So let's consider carrion flowers as an example. Carrion flowers smell like rotting flesh. Sometimes they smell like defecation or poop. This is an example of one stapelia, and here's another stapelia. They often have an opening like this to attract the carrion fly. The example we'll talk about in a little bit more detail is not stapelia, but is instead aristolochia, commonly known as the Dutchman's pipe. Here is a cross-section of the flower. Here's where the stamens and the stigmatic surface are, so the male and female parts. And this vast structure looks like this here. It has a foul smell. The fly is attracted to the inside, so it's almost as if it's flying into an anal opening. The hairs are recurved in one direction so that the fly walks in and has difficulty walking or flying out. On the first day, the fly is attracted with the smell, comes inside, and the stigmatic surface is active, but the anther or male surface, the pollen grains, are not. So this is a time in which the fly walks in, and if it's carrying pollen, as it walks around, it deposits that pollen onto this flower's stigmatic surface. After a day, the stigmatic surface is not receptive. The pollen grains mature, and boom, they explode inside this little cavity here, covering the fly with pollen. At the same time, these recurved hairs dry out, and the fly is able to walk away and walk up to the next carrion flower. The next Aristolochia, with the same process is repeated. This fly that now has pollen grains comes inside another flower and deposits those pollen grains on these stigmatic surfaces. An incredible evolutionary system, one in which the plant has totally duped the fly into carrying the pollen effectively from one plant to the next. There are sex attractants that plants have evolved. In some cases, such as we see in the orchids, the flower mimics another bee, and in fact, often they emit a pheromone that attracts the bee. The male bee tends to copulate with this flower. It's a pseudo-copulation, obviously, and in the process of copulating, either picks up or drops off pollen grains. The evolution here is quite remarkable, with these flowers looking very much like different bee types, even down to the presence of eyes. Aren't plants wonderful? What we're more familiar with are color attractants and nectar rewards. And nectars provide sugar for metabolism, and they often provide vitamins and essential amino acids. So through this coevolution, we see a strong reliance of a plant on a particular vector to move the pollen from one flower to the next. After, as I had mentioned earlier, after the zygote has been formed and the seed is matured, then change in coloration is used as a mechanism for plants to attract an organism such as a bird here to disperse those seeds. The reward for the animal system is the food within the pulp. The seed passes through unaffected by the digestive system, comes out, finds itself deposited on a surface or on the soil surface, with the defecation material being a nutrient-rich environment for germination. A very cool and effective dispersal system. One of the coolest, though, is looking at seed dispersal with ants. Here you see an ant and this slimy mess attached to it. That slimy mess is known as an elaiosome. And the elaiosome is a food resource for ants. So ants come, they pick up both the seed and the elaiosome, haul it off to their mound. In the mound, they eat the elaiosome and discard the seed. The seed is then discarded at the edge of the mound, which again is a nutrient-rich environment. Once again, plants have fooled animal systems either for pollination or for resource dispersal into a resource-rich environment. Through modern agriculture, we've taken advantage of breeding programs, and we have enlarged the size of the fruits that plants had initially evolved for dispersal. We see this in cherries, in oranges, in strawberries, and other fruits.